Thank you. It's an honor and a real pleasure to be here today, in particular the occasion to celebrate David and his accomplishments. What I wanted to do today was to tell you a story about some of the work that we've been doing. Now, my lab at MIT has been interested in trying to understand how do we make the most of measurements from individual cells. We're interested in being able to resolve the heterogeneities in individual cells within a population because we think this gives important information understanding how a distribution in cells may contribute to bulk phenomena, whether this is an immune response or in the context of what I want to talk about today, how to make drugs from biological organisms. Now, we've been interested in a number of different areas, antibody discovery, immune monitoring, but what I'd like to focus on today in the 25 minutes or so that I have here, plus whatever questions you might have, is to really discuss new ways in which measurements on the production of biologic drugs from single cells can give us new insights into the underlying biology of that production at a cellular biology level, and really how this approach can establish a framework for understanding how to focus efforts in strain engineering and optimization with an objective towards improving global access for these kinds of protein drugs. Specifically, I want to look at two things. I want to first examine how integrated single-cell measurements using arrays of sub-nanoliter wells, very similar to things that David has already shown you today, can reveal that the secretory capacity of individual cells is in fact limited in its ability to transport folded protein out of the endoplasmic reticulum, the ER, and that this is really a key kinetic bottleneck in the process of protein production at a cellular level. And then second, I want to consider how we can use these insights to improve our mechanistic understanding of that using simple computational methods for the flux of folded protein through the ER based on simple ordinary differential equations. So two topics today, really. One, looking at how do we make measurements on individual cells that produce drugs and what do we understand now about the biology of that at a cellular level? And two, how do we start to make sense of that in a mechanistic way to begin to think about strain optimization? All right, so problem that we got interested in a few years back was really in how do you make protein biologics very cheaply. And one motivation for this is really quite simple, and it's shown here by these variety of pie charts. What you see here is the global burden of cancer. You can see this on the basis of both developed countries, first world countries like this one, as well as developing countries elsewhere. And perhaps what's a bit alarming about this epidemiological trend is that the burden of cancer in developing countries is increasing faster than that in developed countries. There's a few reasons for this, but you can see as we project out, there is going to be an enhanced burden of all kinds of cancer, even the most common forms that we typically don't think of now in this country, elsewhere. Now, most of you are probably familiar that there are some very amazing drugs available, mostly monoclonal antibodies, that are proteins that are produced in cells and used to treat these types of diseases, predominantly in the first world. Now, I've shown here an example of Herceptin. Herceptin is a molecule made by Roche. It's used to treat breast cancer. This molecule currently costs somewhere between $3,000 and $9,000 a gram to produce. Estimates of how low you can go in cost of goods of production for monoclonal antibodies of this sort can get you down in current systems using Chinese hamster ovary cells and other mammalian systems that are used in sort of platform manufacturing of these molecules. You can estimate maybe $75 to $100 a gram today, and with massive parallelization, maybe you get down to $30 a gram. Now, contrast this to small molecules that are used to treat cancer today. They cost anywhere between 50 cents a gram to Taxol at about $200 a gram currently. So in many ways, we're still about an order of magnitude, if not two, away from being able to make very cheap biologic drugs that would be affordable in a number of different parts of the world. In fact, the target number that we've seen through some of our work with the Gates Foundation has been that $5 to $10 a gram for biologics would be a good number to be able to target for distribution of these kinds of molecules. Regardless of the development cost, just at cost of goods, what could you do in private-public partnerships? To overcome this barrier, one's going to have to rethink the way in which we produce these drugs. And what we've been interested in is using a microbial system that secretes these molecules to try to improve downstream purification, to reduce the kinds of separations that have to be done, also trying to keep the back end of the production clean. And in particular, we've been interested in a yeast organism, Pichia pastoris. Pichia is a methylotropic yeast, that is that it can process methanol as a carbon source. This can be used as an inducer of activity in this organism. It's genetically tractable. There's tools to integrate proteins directly into it. And perhaps importantly, it can also be engineered to produce specific human glycoforms which confer effector functions on the molecules that they make. Now, unfortunately, Pichia currently makes about a gram per liter in best case scenarios of a monoclonal antibody. CHO cells, typically as a comparator in commercial processes, make 10 to 15 grams per liter. So the intrinsic challenge of using an organism like this now to produce drugs at a cheaper price point is that we have to increase its productivity. Now, increasing productivity in bioprocess development typically means improving the overall reactor conditions. That is, that you would formulate the media, the oxygen content, the pH, the chemicals that are present, all of these different elements you would maximally optimize in order to get productivity out of those populations of cells. In Pichia, this has been done, and currently you can get to about a gram and a half per liter using fed batch conditions that optimize as many of these parameters as you can. Now, in mass production of this sort, typically there's a thought. And that is that I started with a clonal cell line to produce this drug. I picked somebody that should be, in principle, genetically identical, and I'm going to use that as a source material to produce everything else. And because it's genetically identical, I'm going to abstract that these cells all behave the same and contribute to product equally. So what I want to tell you today is a story about how we've started to uncover that this is not true using single-cell tools to understand the production of these protein drugs in Pichia. So how do we do this? How do we start to think about this as a problem? So what we're going to do is to take engineered strains, and these are production-ready strains. These are strains that could go directly into a reactor to be able to make quantities of protein. They're going to be engineered to secrete particular biologics of interest. We plate them out and pick clonal ones. So these should be genetically identical. The background mutation rate in Pichia is very low, and we don't expect spontaneous mutations in these populations. We can go through rounds of plate colony purification. And then for most of the work I'll show you today, we've done in shake flasks. And what we can do then is to vary the conditions of production and extract cells from that reactor in real time and sample them using an array of sub-nanoliter wells. It's very similar to the etched arrays that David showed earlier in the fiber optics. Here, the chip is made out of polydimethylsiloxane. And on this chip here, there's about 85,000 containers, so the equivalent of about 1,000 96 well plates. Each well is about 100 picoliters in volume. Now, because of this very small size, we can do things now to make measurements on these individual cells that you might not otherwise be able to do in bulk. One example of this is a process that we call microengraving, wherein we can take a glass slide that is coated with capture reagents, in this case, antibodies directed against the proteins of interest being made by the yeast organism. But in principle, this can be a variety of things, including antibodies against cytokines, antibodies, or other analytes of interest. What we can do is temporarily close a glass plate with that coating against the array so that each cell now sits in its individual reactor of about 100 picoliters. If we allow this to evolve in time, the cells will secrete material into that solution and will be captured onto that other surface. And so after a short period of time, we can separate the two plates. It's non-destructive. On the one hand, what we get is a protein microarray, where each element in the array now matches spatially to the array of sub-nanoliter wells. And on the other hand, we retain the cells. They sit in the individual wells, and we can put this back into media to analyze further or to recover cells that might be of particular interest. Now, one of the things about being able to put this into a small container is that we don't just have to now measure whether or not a cell made an analyte or not. The conventional enzyme-linked immunospot assay provides you that type of information, but rather this now confined volume of defined size allows us to do quantitative measurements of secretion from individual cells. Now, shown here is a model wherein we're looking at the rates of secretion of an individual cell. The theoretical line is shown here. Now, we've modeled it here as a continuous process. This may or may not be true, but for the purposes of modeling, it becomes a convenient way to look at what is happening in the reactor. So here we have a cell secreting at a constant rate. You can see initially the diffusion of the molecules is largely in solution. You can see that here in the movie where they're diffusing into space. But eventually I reach a steady state condition where the situation has flipped. And now we have steady state distribution of those molecules reaching to the distant surface with the capture analyte. And so what this means is that we have a window in which we can do quantitative capture. From about 15 minutes out to several hours, obviously at some point we saturate the surface, and at some point you also lose viability of the cells in these containers. But it creates this window in time between about 20 minutes and a few hours where you can integrate now the rates of production over a defined window in time, and with proper standards you can convert this back into a number of molecules. And so what we get then, by measuring at a fixed period in time for all of the containers on the array at once, is a distribution in the rates of secretion that can now be quantitative. So we've fixed the time period. Each cell might produce at different rates, and what we measure then is the distribution. So we get counts of how many cells are producing, as well as how much material they've made. The example shown here is for cytokines released from T cells, but we will use the same principle here today to talk about the production of drugs from these yeast organisms. So what does the process then look like that we want to use to study this? Well, using this array, we can make two different measurements. We can first perform microengraving to capture secreted products on these cells. Now, I should point out that when we sample the reactor of these yeast organisms and we put them onto these chips, we can do so in about 10 to 15 minutes. The average time it takes for a molecule to transit through the ER and Golgi of a cell is about 2 hours. So what we're really looking at is the history of what this cell was doing in the reactor itself. So we first measure the secretion. We get the protein microarray, which we can then look at the rates of secretion for individual cells. We can map that back by image cytometry. We can, in this case, we're counting cells, but we can also look at the average age of the cells. We can follow them dynamically and look at how they bud. A number of different elements that I'm not going to have time to talk about today. But just to point out that we can, in fact, make quite detailed measurements in cytometry about how those cells are behaving and link them back directly to secretion. This integration of data for the single cell gives us the opportunity to really ask questions about how different phenotypic traits are related at the individual cell level. What we can then do is integrate those data. And this is an example of the integration of the rates of secretion for single cells counted on the array, represented now as a heat map in a histogram format so that you can see where the median rates lie. So where we wanted to start then was to just start very simply at the very beginning. It turns out that this organism's been used to produce a number of different molecules of commercial interest, including insulin as well as vaccine components for hepatitis B globally. And there's a number of groups that are interested in making different kinds of molecules in it, whether for reagents in biochemical assays or for production. But there's been very few systematic studies of how this cell produces different proteins. And so what we wanted to do was build a very simple system where we could ask this question in a couple of ways. First, we wanted to make strains that constitutively express proteins, including EGFP, a very simple protein with no post-translational modifications, spontaneously folds, and no disulfide bonds, as well as glycosylated and aglycosylated sections of the back end of these antibodies, so including the disulfide bonds, the back end of this molecule that provides effector function of it. What we did was knock these in and defined the gene copy number by qPCR, as well as measured relative mRNA levels. So we have a very well-defined cohort of organisms now to study this. We do this both under constitutive production as well as the inducible methanol-based production as well. And one of the interesting things to note is that generally this methanol induction is thought to be a very strong promoter in this organism. But interestingly, at the mRNA level, there's very little difference between the constitutive producing system as well as the induced one, suggesting that perhaps degradation of the mRNA transcript following induction by the unfolded protein response is dominant here, or that the AOX is not significantly more active than what has been previously considered from bulk measures. So one of the first things we wanted to do was to ask, how do these cells produce protein? And by doing cytometry, we can look at very simple questions. Using the GFP as a marker, we can say how many of the cells actually make folded protein. To be fluorescent, it must be folded. And if we look in each of these different strains, about 97% of the cells measured make folded protein, and the localization here is consistent with localization in the ER in this organism. So all of the cells, to a large extent, are making protein of interest. The question is now, what do they do when we look at secretion coupled to that? So if we now make single-cell measurements of secretion as a function of copy number, what we see is that we do get a linear increase in the median rate of production as a function of the copy number under each of these different conditions using the constitutive producer. This is important because it says that as we titrate up copy number, we get more protein. This is good, and it also suggests that constitutive production does not in any way limit the overall productivity of these cells. But interestingly, these data only represent about 2/3 of the total population. You can sample this at different points in the reactor. You can sample it under different proteins. You can look at a whole wide range of different qualities of the production, and it turns out that you always have about 65% of the cells making this. Now, CHO cells, which are commonly used as mammalian hosts, we've also looked at this question. In that instance, about 80 to 90% make detectable amounts of protein. In this case, it's about 100 picograms per mil per hour as a lower limit of detection in this particular assay. But there's a discrete population that is not contributing to the overall reaction. Now, if we also look at the AOX promoter, we see a slightly different story. Now, as we titrate copy number, we're actually seeing a slight decrease in the overall distribution, and if not median rate in production, up to the point where we're looking at glycosylated FC, where this is right at the limit of detection of the assay. Very small population, but again about small rates of production, but again about 65% of the population is in that group. Now, just to rule out that this is not an artifact of the way that we culture the cells in a shake flask, oxygenation is important of this organism. We've also done these studies in bioreactors at 5 liters by fed batch, and what you can see are very similar distributions under the constitutive promoter as well as the AOX promoter, and at a bulk level, they do make similar levels of protein, although you do see a definite delay in productivity with this AOX promoter relative to the constitutive production. All right, so what do these measurements tell us about the secretory biology? If we go back to basic biology, one of my colleagues, Harvey Lotish, and his very canonical textbook in molecular cell biology, we know that transcription occurs, translation of that mRNA that's provided from the gene is done into the ER. Here you have folding and initial post-translational modification by glycosylation. Those materials are then transported by vesicles into the Golgi, which is stacked, where it's further modified with additional glycosylation, and then ultimately secreted out to the surface. So this is the process by which these proteins are made. They're targeted for the ER, and so they have a choice of transiting out or accumulating here in the ER, and there are degradation mechanisms at play here. And one of the reasons that Pittcon is used in cell biology actually as a model organism for studying mammalian secretion is that it has a stacked Golgi apparatus, unlike Saccharomyces cerevisiae. So this becomes a very efficient process, and one in which it seems reasonable that we should consider how this might work for the production of proteins. Now, I've shown you these distributions, and if we take these distributions, assuming that they're Poisson processes for secretion, we can look at how to model the overall productivity. We can fit these with gamma distributions with 2 parameters, B and A. Now, physically, given these types of measurements, we can interpret B and A as meaning roughly the mean rate at which vesicles are being released to the surface, really from the ER transiting through the Golgi to the cell surface, and the mean size, or the number of molecules that are packed into those vesicles. And so now, as a function of copy number, we can look at how those 2 parameters trade off. If we look now at burst frequency versus burst size, as we increase gene copy number, we see that there's a decrease in the number of trafficking events, but an increase in the overall burst size. We're making more protein, they're packed more full, and they are being released. And this is true under the FC region as well as the GFP, with a shift, of course, in the overall productivity, as I've already shown you. Interestingly, though, we do not see any examples out in this region. Right, so it says that this inverse relationship suggests that there is, in fact, a fixed capacity of these cells to be able to produce secreted protein, and that there's a discrete set of components for shuttling that protein. So if we look at this a little more closely now, we go back to the ER and think about what happens. showed you that most of these cells have folded protein, which means that it's made it into the ER. Once you're in the ER, you have a choice. You can either be transported out through the Golgi, and the kinetics of transport here have been well studied by people like Jennifer Lippincott-Schwartz, who have looked at these types of processes by microscopy. A competing process, though, is ER-associated degradation that's associated with proteasomal degradation, the unfolded protein response, in which protein that's accumulated can be shuttled out in this particular way. So protein comes in, it folds, and it can either go productively to transport out, or it can be degraded. So now, by this integrated measure that we can do, where we can look at intracellular content versus our secretion measurements by microengraving, we can actually do one-for-one correlations now of intracellular amounts to secreted protein. And what's interesting now is here you can see these two populations that I showed you earlier in pie chart form, now measured at the limit of detection here of the measurement for both secreted GFP as well as intracellular GFP. You can see there are two distinct groups that coexist here. Now, and this is true also under the methanol-induced promoter as well. One of the important questions is, is transcription and translation a rate-limiting step in the kinetics of all of this? So to test that idea, we created a strain that made both GFP and glycosylated Fc. What we would expect kinetically, if transcription and translation was a rate-limiting step, would be no correlation between the rates of secretion for these two proteins. Instead, we see a very strong correlation of these, scattered by extrinsic noise suggesting different cell capacities for secretion amongst these individuals. And for the aficionados, these are in fact put into two different loci in the organism. And so what we do not see, though, is this kind of acorrelated behavior you would expect if transcription and translation is the rate-limiting step. Also remember that eGFP as well as Fc are very different proteins in the amount of time required to fold and transit through the secretory pathway. All right, so given this observation and given what I've shown you about the populations, we went back and we modeled this transport process using simple ordinary differential equations to describe the accumulation of folded protein in the ER, governed by three parameters: translation, transcription, secretion or production, and then degradation or sequestration of the protein. And if we model this now for these populations of cells, what we find is that under different conditions we see different relationships between intracellular protein and protein secretion. When the dominant process is degradation, we see this positive correlation between protein secretion and intracellular amounts. And when secretion is dominant, we would expect an inverted relationship. Now recall I showed you earlier, we do in fact see a slight positive correlation, suggesting that we're mostly in this domain. So that does in fact suggest that the steady state production of protein from these cells is done so under a condition where the degradation is more dominant than the productive secretion. We can look at the model further to try to figure out where is it we would want populations of cells. Suppose we want high secreting cells, ones that are productive. They should sit out here in the distribution, high rates of relative protein production. If we look at that population, what we see is that in fact they should have high rates of secretion relative to ERAD. So we want to be able to find those individual cells. Now, I showed you these distributions. If we target the cells that we see at the tail end of that distribution, it has a known zip code and street address. And so what we can do is go back to those individual cells and recover them. We do this by automated micromanipulation. We feed in the address of the cells. Using an automated capillary system here, you can drop into these individual wells, recover that cell, and then transfer it back into a conventional 96 or 384 well cell plate. And so in this way, we can recover cells from different parts of the distribution to ask how stable those populations would be. Now, if we do this experiment, we can target in these distributions high secretors, intermediate secretors, low secretors, and even the non-secreting populations. If we take these individuals and we then grow them out for several doubling cycles over 24 hours, we find that they're really no different than the parent strain. That is, given that there is a very low genetic mutation rate in this organism, we can't get something for nothing, and that these protein secretory states are intrinsically unstable. This has implications in selecting master cell lines used for manufacturing, because it means that as you select out at the tail end of these distributions, you may not be selecting on somebody that is truly a stable producer, but rather somebody that is an enhanced state relative to its normal distribution. It's oftentimes that you take lines into mass production that start to decrease their performance over time, and we would suggest that epigenetic variance of this sort is a contributing factor to that. So you can't get something for nothing. Let's do better. And finally, just to kind of close out this idea, now we have to introduce something. So by chemical mutagenesis, we're going to go in and do this experiment again, now targeting these guys, but under now genetically diverse conditions, recover those individuals, grow them up, and do this through several rounds. If we do this, in fact, what we see is that after 3 rounds, we do get improved productivity from the pools of these strains, and now we can recover individual cells and see how they perform. And I think what's exciting is that, in fact, now we can find better populations. These are clones that have now been tested as many as 2 years out from the freezer, and they do show enhanced productivity at about 2.5-fold increase over the baseline of the parent strain. In this organism, reported increases of 1.2 to 1.5-fold are common and considered to be large. So the fact that we can find these larger producing cell lines is quite important. Given that in the same population we have lower producers, this also gives us an opportunity to think about how to understand the improvements. Now, a long time ago, well, a long time by my generation, you wouldn't have been able to do this kind of forward genetics experiment. You would have had to do this experiment and you would have said, well, that guy's a better producer, but I'm going to have no idea why. But thanks in part to David and to many others, with the advances in next generation sequencing technologies, we can go back and resequence these guys for about $400 a strain and do comparative genomics and map out the lineages of how these cells relate to these cells and to the parents from which they were derived to start to identify the key genetic factors that are driving this higher level of production. And as much as I'd like to tell you that story today, I won't have time, and it's still in development, but I think it is just another indication of the kinds of transformational work that David has been a part of in his career. So to summarize here in the last couple of minutes, I've shown you that single-cell measures show protein trafficking through the secretory pathway is rate limiting for this organism. This is an intrinsic bottleneck now of using this kind of organism or any other like it in its family as a way of producing these protein biologics. They're very cheap to culture and grow, but we must overcome this bottleneck to drive down production cost. What I've shown you is that increasing the complexity of the proteins also reduces the median single-cell productivity. This is another point as we think about making full-length antibody. It will be important to optimize and to perhaps do these types of clonal selection with mutagenesis strategies that I've shown you under optimized conditions with large copy numbers of antibodies. The secretory states are inheritable, not stable. Some work that I didn't have a chance to tell you about today was that we've shown at a single cell level that these states, that cells can flip back and forth between on and off production states over short intervals, that they're inherited by daughter cells, but that over long periods, as I did show you, those states revert back to the normal distribution through many rounds of division. The mutagenesis and screening strategies do in fact yield better strains, and that with the advances in next generation sequencing, we can really start to do forward genetics to improve rational strain design of this organism. Some of the implications of the data that I've shown you are that targeting components involved in transport from the ER and Golgi and out of the cell are critical, that it's in this area that enhancements need to be made. We think that there's a need to develop new strategies to control ERAD and protein degradation or sequestration. That typically these have been upregulated as a way to enhance protein production, but it's really the balance between transport and ERAD that's most important. You should identify and modulating regulatory elements that govern the abrupt transitions between secretory states may also be important because you may want to reside in productive states longer. And then finally, engineering promoters, which is commonly done in these types of systems without also addressing the secretory capacity, should only yield modest gains, and that's been played out in the literature multiple times now. So I just want to finish by thanking the folks that have been involved in the work. Sanger and Bog is a postdoc in the lab that's really been taking on the forward genetics and as well as trying to understand the mechanisms behind these things. Tim Politano developed the modeling framework that I showed you. Vasiliki was a MS student in the lab that performed the first rounds of screening on the mutagenized sets. And then really this whole program's been largely led by Carrie Love, who has really taken us into this area in collaboration with Glycophi and the KI Bioinformatics. Now, as the last speaker, it's hard to say anything new that other people have not said about David, but what I wanted to do is to summarize what I think the common theme of this session might have been. And it really harkens back to a particular quote that's right up front on David's website. That is, our laboratory investigates new ways to measure things. I think that that as a theme is something that all of us here can appreciate, and that I think many of the different talks here today have hit on a variety of different ways in which you can make measurements. So thank you, David, and congratulations.
Thank you, Dr. Lowe. Any more questions?
How stressful is your physiological condition of the single cell? In other words, can we use it on CHO cell line?
Yeah, so we've done these measurements on CHO cells as well. We think that the short-term nature of the measurements is not strongly perturbative to the system. In fact, we do a lot of work in primary cultures of T cells, which are much more sensitive to these things, as well as cancer cells. And we find that in each of those instances, we can do assays out up to a week in these conditions. So the very short measurements that we make is not strongly perturbative enough to substantially change the populations.
And if you can select a winner from the CHO cell line, for example, can that still be developed into a master cell line?
Yes, we have not spent a lot of time doing that, but we have cloned out CHO cells in the same type of process. Fran?
This is kind of a mundane question, but where you showed the capture antibodies on your top plate, those could also be variable in your chemistry of a mobilization, could be variable over a large quantity, and that could bias your results. How do you deal with that?
Yeah, growing up as a surface chemist, it kind of makes me cringe that we do this, but in fact, we do just distribute them over the surface, and our kind of understanding of the distribution of the antibodies on this is that over the kinds of surface areas that we measure, roughly 50 by 50 microns squared, that we get a pretty uniform distribution there to the best that we understand it, but at the resolution which we read the information, at micron optical resolution, we think that it averages out over that. So on a given experiment, we would average something like 2,500 pixels of information. So it's fairly reasonable, although the point is well taken. David?
So what is it about the inside of the well, or the medium, that causes these cells to get stuck, but then they get unstuck when you start growing?
With respect to the production?
Yeah, some are non-secretors and some...
Yeah, so I think what it really points to, and we've seen these data now also in a variety of different forms, CHO cells and T cells, is that secretion is much more regulated as a biological process than we've typically appreciated. I think that at least in this community, when you express protein, it should just be coming out of the cell. And so I think that these results have been quite startling to this as a community, is that in fact, no, there are people that are just along for the ride. No, the fact that we see them switch back and forth also suggests that there's probably a stress response, and we know that this organism constitutively has the unfolded protein response upregulated. And so it may be that some balance of that as a process is allowing them to switch digitally back and forth to the point where you get complete shut off for a period and then fluctuation back in.
Any more questions? I want to thank you for all the speakers for coming and present these valuable research results. And also thank you all for attending. Last but not least, thank you and congratulations to Dr. David Walsh. And thank you so much.